Um, Hello everyone. Welcome to today's Museum seminar. It's really my pleasure to welcome our today's speaker, Dr. Liu Dan from Westlake University. So Dan is a fresh PI. I think she just started her lab a couple months ago. So, and but I think she has a really astonishing CV. So Dan received her bachelor degree from School of Life Sciences at Wuhan University in 2011. And then after which she got her PhD from the Department of Basic Medical Sciences at Tsinghua University in 2016. So after she finished her PhD training, she actually moved to US from 2016 to 2023. Then actually joined Howard Hughes Medical Institute and Department of Microbiology and Immunology at UCSF in Jason Sister's lab as a postdoc fellow. So she, after 7 years training, she actually joined School of Life Science at Westlake University as in 2023 as assistant professor in the lab of dynamic immunology and imaging. So one of the things, so I think Dan actually get a really restrict and restrained training, both her PhD and the postdoc, which she actually utilized different technology, particular imaging technology, to study B-cell biology, and which I think particular I get super impressed is on her CV, her PhD training. She pretty much had 4 publications, 2 with first author as Nature and Immunity, and then the postdoc training end up with 5 papers, 4 first author and co-first author paper on Science, Nature, Immunity, and JXMed. I think she got a lot more paper than I actually start my own lab for these years. So I think that's pretty impressive. After she actually started her program, I think she, as I mentioned, she has made a really important discovery in research of immune cell differentiation and its role in humoral immune responses. Her lab plans to integrate classical immunology, computational biology, and the CRISPR technology in vivo screening and intravitreal to photon microscope in mouse model and human samples in order to study and understand how immune cells interact with their tissues as with surrounding microenvironment to maintain homeostasis, as well as how dysregulation of these processes leads to immune disorders and diseases. So I think her research program potentially can allow for regionalized regulation of cell and provide new perspective for vaccine design and immune modulatory therapies. And then their researching interested also potentially can focus but not limited to other directions, which including molecular machinery and function of interactions between immune cells and their microenvironment, development and the function of myeloid cells, which including dendritic cells, monocytes, and macrophage, and also immune surveillance against autoimmune disease and tumors. So as I just mentioned, with all these really incredible achievements, she is recipient of Cancer Research Institute Fellowship and the Wu Prize. So I think a lot of us actually probably already know she just has a new study on Nature Immunology published probably just a couple of weeks ago. And I think we're gonna hear a lot about this study regarding the red blood cells trigger splenic marginal zone B cells, retention and the function. So without further ado, then the floor is yours. We are looking forward to your talk.
Thank you so much. Thanks so much for invitation and a very nice introduction, and give me the opportunity to present my recent work. The main focus of my talk today is about how marginal B cell interact with the surrounding red blood cell to maintain their homeostasis and functionality. So the spleen, as the largest secondary lymphoid organ, plays a crucial role in protecting the body by preventing the passengers from reaching the mucosal barrier and entering the systemic bloodstream. So according to the spleen section with the marker TR119, we can see the spleen is filled with a red blood cell, and this region we call as the red pulp. So, and we can imagine that the spleen is the immune organ rich in the in blood flow. And then the region with a high expression of the IgD is the B cell zone, and the area surrounded by the B cell zone is the T cell zone. So this area where B cell T cell are located constitute the white pulp, and the interface between the white pulp and the red pulp is known as a margin. In the image, the green cell, the green the band-like area, the green region is the margin. So blood passes through the margin from the marginal sinus and then reach to the red pulp, finally return to the circulation by the venous sinus. So the marginal B cell, the marginal cell are rapidly exposed to antigen in the bloodstream. Marginal B cell constitute a major cellular population in the marginal area. Compared to the follicular B cell, marginal B cell have a high expression of CD21 and the IgM, and the low expression of CD23 and IgD. So when we use the PE conjugated antibody for brief labeling by IV injection into mice, the cells located in the blood exposed region can be labeled within 3 minutes. So typically about 55% of the marginal B cell can be rapidly labeled. So in the spleen section, this labeled cell are turned yellow. Marginal B cells are not stationary in the margin, the shuttle between the follicular region and the margin region. So during the antibody injection, the remaining cells located in the follicular region are protected and remain unlabeled. Marginal B cell also can recognize the antigen in bacterial membrane, and once antigen are recognized, they can rapidly generate plasma cell. So the unique location of this marginal B cell directly exposed to the flowing blood facilitates their encounter with the circulating antigens, leading to the rapid production of antibodies. So for the human spleen, it received about 5% of the cardiac output, a substantial blood supply for an organ with no high oxygen consumption at baseline. So the degree of the perfusion in the spleen evidently contribute to optimal immune surveillance and the clearance of the particles and the cells in the blood. So in the human spleen, there are also marginal B cells characterized by the IgM positive, IgD positive, and the CD27 positive. But their shape a little different from the marginal area in the mice. So in the human spleen, the margin surround the follicular zone, but even some difference in the organization, but a certain region of the margin still directly expose themselves to the open blood circulation. The development of the margin B cell are relatively slower compared to the follicular B cell. So immature marginal B cell may be one reason for the poor immune responses to the certain T cell independent antigen in newborn mice and human infants. Also include adults who have the splenectomy surgery. This kind of patient will require antibiotics to prevent the chronic infection. So So currently, there are several signals have been discovered to regulate the generation and the positioning of the marginal B cell in the spleen. So members of the TNFR family maintain their survive. Integrins enhance their adhesive abilities, and the BCR signal regulate their environment. And the S1PR1 control the shuttling of the marginal B cell between the marginal and the follicular region. So even we already have some understanding of the localization and the homeostatic requirement for the marginal B cell, but considering about the specific localization of the marginal, the mechanisms by which marginal B cell perceive their specific position relative to the red blood cell and the blood flow are not fully understood. So in order to observe the real-time motility of the marginal B cell in the marginal, so here we use the intravital to photon microscopy imaging, try to observe the real-time interaction of the motility of the marginal B cell. So here we transfer the GFP positive B cell into the CD19 knockout mice, because the CD19 knockout mice have no endogenous population of the marginal B cell. So after 8 to hours weeks reconstitution, the transferred B cell began to occupy the marginal, and finally the majority of the transferred GFP B cell show the CD21 high, CD23 low marginal B cell phenotype. And then within the initial 3 hours of analysis, we infused mice with a red blood cell labeled with the PKH26. Finally, we can get 2% of the red blood cell labeled in the recipient mice. And also, we will transfer the cell trace violet labeled the follicular B cell into the recipient mice to help us identify the lymphoid follicle region. So then we scan the different region of the spleen, try to find the identical the GFP labeled marginal B cell area. Here we can find some area like the imaging shows the GFP positive marginal B cell clustered near the CTV, the blue labeled follicular B cell. And we can envision the marginal like the 3D structure, like the spherical structure. Then we scan from the surface to the depths used to photon microscopy. We observe that within the increasing depths, most of the green B cell surround the blue follicular B cell. So this indicates that in this recipient mice, all marginal B cells are fluorescently labeled with a green color. Then we collect some movie, try to observe the real-time motility. So first, we can observe that in the certain area of the marginal, some red blood cells flow uniformly through the marginal, but in the other area, the red blood cells seem to move in an irregular manner. And also, we can see that the red blood cell passing near the marginal B cell we can observe some this B cell, and also we can see some transit contact between the marginal B cell and the red blood cell. So here I use the blue arrow to point some interaction between the red blood cell and the marginal B cell. So for some case, we can see the red blood cell come from the blood flow and then come to interact with the several different marginal B cell. And finally, they will lost into the blood flow again. And then some cases show the red blood cell just come from the flow and stop by the interaction with the marginal B cell. Then with a very transit contact, then they also will go back to the flow again. So here is another example. I track the movement of the several red blood cells and mark them with the yellow lines. And here, the blue arrow indicates the contact surface between the marginal B cell and the red blood cell. So we can see some interaction involved a very light surface interaction, but some others involve a relative larger area of the entangled interactions. So according to the two movies, we can easily detect contact between the red blood cell and the marginal B cell in the 20 to 30 minutes video. But I want to emphasize here, because the label with only 2% of the red blood cell are labeled in these movies. So in the reality, the contact frequency should be expected to be 50 times higher than shown in the movies. So this data suggests that in the flowing area within the marginal B cell frequently come into contact with the red blood cell. Because the observed frequent interaction, we want to investigate whether there might be a ligand receptor pairs on these two cell types that could be involved in the function of the marginal B cell. But because the red blood cell is very specific cell type, there are very few surface molecules expressed on the red blood cell. So here we find the CD55 is highly expressed on the red blood cell, and it is the one ligand for the CD97. And by the flow cytometry, we find the marginal B cell highly expressed CD97. CD97 is known as the adhesion G-protein coupled receptor, and they have a NTF and CTF two fragment, and within the two fragment, they have one autoproteolysis site. And once the ligand bind to the receptor, the NTF and the CTF associate non-covalently through this domain. And after the outer force applied to the NTF to remove the NTF, then the G-protein coupled receptor will be activated. And because I mentioned before that the marginal B cells are located in the blood-enriched region, so it indicates they may have an outer force like shell force to apply to the NTF or the CD97. So based on the high expression of the CD97 and its mechanical properties, so we investigate the function of the CD97 on the marginal B cell. So we analyze the CD97 deficient mice, and we find a significant reduction in the number of the marginal B cell, and the immunofluorescence staining of the spleen for IgM and IgD also showed a decreased thickness of the IgM high marginal B cell in CD97 deficient mice, consistent with our flow data. Because here we use the full body CD97 deficient mice, so all cells from these mice are without CD97. So in order to check whether the absence of the CD97 in B cell lead to the deficiency of the marginal B cell, so we employ a bone marrow camera system. So we mix the 50% of the CD45.1 wild type 41 positive wild type bone marrow with the 50% CD45.2 positive wild type or CD97 deficient bone marrow. And in the chimeric system, all cells share the same microenvironment. Then here we use the CD45.1 to gate the CD45.1 positive cell and the CD45.2 cell, and then we check the ability of these two population to form the marginal B cell. So in the wild type camera, we can see the marginal B cell percentage from the CD45.1 wild type and the CD45.2 wild type have no difference. So that indicates the ability of the CD45.1 and the 0.2 cell to form the marginal B cell is similar, but in the knockout camera, the percentage of the marginal B cell produced by the CD97 deficient B cell is significantly reduced. This suggests that the B cells themselves require CD97 to maintain their homeostasis of the marginal B cell. To further explore the reasons of the loss of the marginal B cell, we compare the development, proliferation, and the apoptosis of the CD97 sufficient and the CD97 deficient B cell, but we didn't observe any significant differences. So finally, we hypothesized that reduce the frequency of the marginal B cell in CD97 deficient mice might be the result of their migration into the bloodstream. So through the two-photon microscopy, we observed some CD97 deficient GIP positive B cell were indeed lost into the blood circulation, because from the imaging we can see in each time frame the number of the cell lost into the blood is very few. So we try to use antibody to block CD97 and make the cell release more synchronized. So we find that when the mice were treated with the CD97 blockade antibodies, more marginal B cell could be detected in the blood 3 hours later. Then in order to track the fate of the marginal B cell already released into the bloodstream, so we intravenously transfer the GIP positive splenocyte into the blood. Then we can see the percentage of the marginal B cell in the donor spleen was about 8%, but 10 minutes after injecting these cells into bloodstream, the percentage of the marginal B cell detectable dropped to 0.5%. This suggests that once the marginal B cell entered the blood, the majority of the cells lost their phenotype of the marginal B cell. And at this point, the homing GIP positive marginal B cell to the spleen gradually increased, reaching a steady state within 6 hours. But when we transfer the CD97 deficient GIP splenocyte, we can observe a similar loss of the marginal B cell in the blood within 10 minutes. And also we can see the initial appearance in the spleen and the peaked in homing at 6 hours. But these cells were not sustained. The majority of the cells had disappeared by 18 hours. So the reduced ability to repopulate the spleen is also an important factor contributing to the overall defect in splenic marginal B cell in CD97 deficient mice. So according to my previous study on dendritic cell indicating that the CD97 can transmit signal through the G13, so we wanted to investigate whether the function of the CD97 in a marginal B cell requires G13. So we also checked the mice with a selectively deficiency of the G13 on B cell. We can see a twofold reduction in the frequency of the marginal B cell similar to CD97 deficient mice. And when we also use the CD97 blockade antibody to treat the G13 deficient mice, we couldn't see a further reduction of the frequency of the marginal B cell. But when we treat the wild type mice with the blockade antibody, as expected, we can see the reduction of the percentage of the marginal B cell. So this data confirm that the CD97 and the G13 are in the same similar pathway. And the RGF1 is one of the most specific downstream effector of the G13. So we can see the similar loss of the marginal B cell in the RGF1 deficient mice. So these findings suggest that CD97 signals in the marginal B cell by the G13 and the RGF1. So earlier I mentioned that CD55 is the validated ligand for CD97 in vivo. So to test whether CD97 depends on CD55, we also analyzed the CD55 deficient mice. We can see the similar reduction in the frequency of the marginal B cell in the CD55 deficient mice. And the CD55 is broadly expressed in the cell originated from the hematopoietic system and the non-hematopoietic cells. So to determine the cell type expressing CD55 that are necessary for the homeostasis of the marginal B cell. So we generate a different type of the bone marrow chimeral. Stroma cells are very resistant to the radiation, but the hematopoietic cells are very sensitive. So here we irradiate so we transfer the control and the CD55 deficient bone marrow into the wild type lethal irradiated recipient. And we can see the marginal B cell show the defect. But when we transfer the wild type bone marrow into irradiated control and the CD55 deficient mice, the marginal B cell compartment is, we didn't find any change. So this data suggests that the CD55 on bone marrow derived cell is involved in maintaining marginal B cell in the spleen. Follicular B cell express high level of the CD55. So to determine whether the CD55 on B cell itself is involved in the process, we also generated a bone marrow chimeral. So we mix the CD45.1 bone marrow, wild type CD45 bone marrow with the CD45.2 CD45.2 positive CD55 deficient bone marrow. Then we find the CD55 deficient B cell have no disadvantage to form the marginal B cell. So we can see the CD55 on B cell doesn't play a role in maintaining the marginal B cell. But if we mix the CD55 deficient bone marrow with the CD97 CD55 double knockout bone marrow, so CD55 deficient B cell have a CD97, but the double knockout a cell have no CD97 CD55. So in this chimeral, in the whole environment, there's no CD55. So in this environment, no matter the B cell express CD97 or not, both of them couldn't maintain the marginal B cell in the spleen. So according to this data, we can conclude that the CD55 is the only ligand for CD97 in regulating marginal B cell. Considering that the marginal B cell is in a region enriched in the blood flow and consistently interacting with the red blood cell, as imaging showed, and also the red blood cell express CD55. So we hypothesized whether CD55 on red blood cell directly participate in the regulating marginal B cell. So to test the hypothesis, we purify the wild type or CD55 deficient red blood cell and transfer this red blood cell into the CD55 deficient mice. So after reconstitution, finally we can get 65% of the red blood cell derived from the donor. So with the transfusion of the wild type red blood cell, we can see the defect of the marginal B cell in the CD55 deficient mice could be totally rescued. And the platelet also express CD55. So to test the potential contribution of the platelet CD55 to the homeostasis of the marginal B cell, so we also use the chimeral system to mix with the MPL deficient and the CD55 deficient bone marrow, because the MPL knockout mice cannot produce the platelet. So in this chimeral system, all platelet were without CD55, but the other cell type express CD55. So finally, we find the deficiency of the CD55 on platelet didn't lead to any reduction in the marginal B cell compartment. So the red blood cell are a crucial CD55 positive cell type required for the homeostasis of the marginal B cell. So when we check the CD97 expression from the CD55 deficient mice, so surprisingly, we find the CD97 is elevated in the CD97 is elevated in the CD55 deficient mice. And this elevation was restored to the normal level after transfer of the wild type red blood cell. So this suggests that the regulation of the CD97 depends on the involvement of the CD55. So as I mentioned before, the CD97 has a self-cleavage site, and when the ligand binds to the receptor, they will have the autoproteolysis. Then the NTF and the CTF form the non-covalent bond, and NTF will be likely can be removed by the outer force. So in order to check whether the NTF was removed by shell force in the blood, we try to mimic the process. So we co-culture the wild type CD55 wild type or CD55 deficient red blood cell with the splenocyte, and we used a shaker to mimic the shell force. Then when we co-culture the wild type red blood cell with the splenocyte under condition of shell force, the extracellular domain of the CD97, the expression level was decreased. But when co-culture with the CD55 deficient red blood cell, the extracellular segment of the CD97 didn't change even under shell stress, under shell force. So then we introduced a mutant. The T419G mutant is a non-cleavable mutant. So when we overexpress this mutant on the marginal B cell, then the marginal B cell with this mutant didn't show any change in CD97 surface staining when co-culture with the wild-type red blood cell under shell force conditions. This further suggests that the decrease in wild-type CD97 is due to the removal of the NTF. And then we overexpress the T419G mutant in the CD97 knockout bone marrow. Then the loss of the marginal B cell couldn't be rescued. But if we overexpress the wild type CD97, we can completely rescue the loss of the marginal B cell. So this indicates that the self-cleavage and the CD55 mediated NTF removal are crucial for CD97 to function properly. Human spleen marginal B cell also express high level of CD97, and the red blood cell also express CD55. So when we co-culture the human splenocyte with the human red blood cell, we also can see that on the cell stress condition, the surface staining of the CD97 was significantly reduced with the shell stress condition. So this data suggests that the human marginal B cell also undergo the removal of the CD97 NTF in interaction with the red blood cell under shell stress conditions. And this conservation might give us the suggestion that our findings in the mouse marginal B cell are relevant to ensuring the efficient surveillance and the response to the blood-borne pathogen in the human spleen. So in order to explore how CD97 enable the marginal B cell to resistant to the blood flow, so we try to overexpress CD97 and its activated form fused with the GFP protein to monitor the effect of the CD97 signaling on the morphology of the 293 T cell. So when the cells when the 293 T cell were overexpressed with the wild type or T419G mutant version, we can see the similar shape, the irregular shape of the 293 T cell. But when the 293 T cell overexpress the activated form, we can see these cells looks more round and looks more small. And also the GFP is a fusion one, so we can directly to see the CD97 localization according to the GFP signal. So we also find in the CTF, the activated form, we can see the very similar typical G protein receptor cluster in the 293 T cell. And then we use the imaging J to compare the maximum diameter of the cell under each condition to confirm the differences in the shape. So we find the cells overexpressing the CTF had the smallest diameter. So according to this observation, we try to imagine the process in the vivo. So we can, so when the marginal B cell extend their membrane into the area where the red blood cell flow with a certain velocity, the contact between the CD55 on red blood cell and the CD97 on the marginal B cell lead to the mechanical removal of the NTF and activate CD97 and the G13 signaling. This associate activation of the Rho A trigger the membrane retraction process, so may increase the adhesion ability mediated by the integrin to the tissue microenvironment. But if the CD55, CD97 signal is absent, the marginal B cell would extend into the blood flow area and eventually will be lost into circulation. To directly test whether the CD55, CD97 interaction leads to membrane retraction, so we use the optical tweezer to mimic the trap force. So we coat the beads with the CD55 and then put the beads contact with the with the CD97 expressing 293 T cells, and then we allowed then we will pull the beads away from the cells. So through the membrane tenders, the displaced beads would be remain connected with the cell and generate a restoring force. Applied to the membrane tension. So this measurement revealed that the retraction force in the cell expressing the wild type CD97 was nearly twice higher than the T419G expressing cells. So this indicates that cells, if the cell express the CD97, they will exert a reactive force detected by the optical tweezer. Then we also co-express activated Rho A biosensor and the CD97 in 293 T cell. So we use the same method, and we also put the CD55 coated beads contact with the cell membrane and try to pull the beads away from the cell. Then the blue arrow indicate the time frame I pour the beads. So we can observe the activation of the Rho A, the blue signal here, at the beads contact point. And but when the cell overexpress the non-cleavable mutant, we cannot find any Rho A activation even with the pulling force. And then here we try to use a timeline correlated with the activation signal of the Rho A. This data suggests that CD97, when bounded to the particles expressing the CD55 and activated by retraction, will initiate a membrane retraction through the Rho A signaling. Marginal B cell can generate rapid antibody responses against the antigens independently of the T cell. And the TMP-FICO is widely used as an immunological model that doesn't rely on the T cell. So here we inject the TMP-FICO into the mice, and after 40 minutes, we check the uptake of the TMP-FICO in the CD97 sufficient and deficient mice. There is a lower quantity of the TMP-FICO binding to the marginal B cell in the CD97 deficient mice, and this defect also contribute to the absence of the B cell themselves, because in the mixed bone marrow camaro, the CD97 knockout marginal B cell still show the lower binding to the TMP-FICO compared to the wild type B cell. So to test the impact of the B cell of the CD97 deficiency on the antibody responses, we also injected the TMP-FICO into wild type or CD97 deficient mice. And after 5 days, we used the ELISA to measure the level of the IgM antibodies against the TMP in the blood. So here we can see the CD97 deficient mice show a reduced IgM response to the TMP FICO. And then also this is the full knockout. So then we also generate the mixed bone marrow chimeral, try to check if the reduction of the antibody response is intrinsically. So we generate the heavy chain A and heavy chain B mixed bone marrow chimeral. Then we can use the antibody anti-IgM-A and IgM-B to distinguish the antibody derived from the wild type or Sigma-7 deficient B cell. So then we find in the IgM-A, all IgM-A cell are wild type. So there's no difference of the modern B cell produce the TMP specific IgM-A. But for the IgM-B, the Sigma-7 deficient knockout cells show the reduction production of the TMP specific antibody. So the Sigma-7 is necessary for the early IgM response to the blood-borne passenger in the T cell independent way. So finally, we want to summarize our findings. The modern B cell extensively migrate within the MAR zone exposed to blood, but rarely exist into the bloodstream. So here we observed for the first time that the modern B cell engage in the frequent interaction with the passing red blood cell, and using the CD55 Sigma-7 ligand receptor system to sense their contact with the bloodstream. So passengers of the CD55 positive red blood cell trigger the CD97 involved in the modern B cell, lead to the retraction of the NTF, then activate the receptor. So in the absence of the Sigma-7, CD55, or the downstream 13 RKF1, the modern B cell in the spleen will lose the ability to circulate and unable to be maintained. And also we use the in vitro optotwizzle system. We find the Sigma-7 can lead to the Rho A activation and the cell membrane retraction. And we also find that this also, we find this system is crucial for the spleen's ability to produce the IgM response against the T cell independent antigen in the blood. Then from my previous research, it's naturally lead to the association that beside the spleen, there are also many other organs, especially in the lung, bone marrow, and the liver, even specific region of the kidney. They're also abundant in blood flow. So here we use the textron to label the blood vessel, and also use the two-photon microscopy to observe the real-time blood flow in these organs. So this will raise a question about how cells maintain their homeostasis, and what's the difference between the cell in contact with the blood flow or not in contact with the blood flow, how the blood flow educate these cells. I think is the topic worth studying in the future. Finally, I want to thank my mentor, Jason, for his support during my postdoc training. I also want to thank the Ben for helping me with the optotwizzle experiment. Also, our lab is growing, so we also thank my lab member. And yeah, I also want to thank the Westlake University and the Westlake Laboratory for their support. And thank you for listening. And once again, thanks Chuan for the invitation. And I hope to use the opportunity to meet more colleagues and establish collaboration. Thank you.